Hi everyone, today we're going to be going over the origins of life on Earth. In this video, I'll be talking about how molecules which are necessary for life today likely first evolved. We'll talk about the main hypotheses regarding the environment in which life evolved. We'll talk about the taxonomy of life on Earth today and what kinds of organisms likely evolved first. And then we'll get into what the fossil record tells us about early life on Earth and how it evolved into eukaryotes and more complex life and eventually us. So on Earth today, we know that life only comes from other life. So how did life start? Two major hypotheses about where life started environmentally on Earth have been made, and these include the warm little pond hypothesis and the hydrothermal vent hypothesis. However, before we get into where life was made, first we have to talk a little bit about the biomolecules that are necessary for life today and how they might have been synthesized abiotically. The four major classes of biomolecules, aka necessary molecules for life today, include proteins, lipids, carbohydrates, and nucleic acids. First, we'll talk about proteins. Amino acids which make up proteins were produced by Miller and Urey abiotically in their experiment in which they ran an electric discharge simulating lightning through a closed system containing the components that would have made up the atmosphere at the time of early Earth, in addition to water simulating the ocean. The amino acids that were synthesized abiotically in this experiment were then used by a scientist named Sidney Fox to form long chains of these amino acids, aka proteins, in another abiotic experiment, showing that not only amino acids but even simple proteins can form abiotically on Earth. And that is likely how the first proteins did form. However, there's another hypothesis called panspermia, in which people hypothesize that amino acids and maybe proteins and other organic molecules came from extraterrestrial sources like meteorites, which seeded Earth with life instead of life actually evolving first on Earth. And so this is an interesting hypothesis. However, for the purposes of life's origin on Earth, it's kind of unparsimonious. We don't really need it to explain amino acids and protein formation abiotically, since we already know by Miller, Urey, and Fox that these molecules can form abiotically without meteorites. The next biomolecule that we'll talk about is carbohydrates. Carbohydrates, or sugars, which are types of carbohydrates, were likely abiotically synthesized in the widely accepted foremost reaction, in which the polymerization, or just forming long chains, of formaldehyde in the presence of calcium hydroxide yields a complex mixture of sugars, including ribose, which is an important sugar we'll talk about later when we get to the nucleic acid biomolecules. And the fragmentation of certain products down the chain of this reaction can regenerate one of the first products of this reaction, which is glycolaldehyde, leading to an autocatalytic cycle, or repeating cycle on its own because it's reproducing some of the products that began its reaction. Additionally, products of this chain reaction can then go on to form higher sugars by reaction of the tetroses and pentoses, so 4-carbon and 5-carbon sugars, with our original reactant, formaldehyde. Moving on to lipids, abiotic formation of fatty acids and glycerols, which are key components of lipids in all of our cells' membranes, which we'll talk about their structure in a second. These have been demonstrated to abiotically form due to dehydration or condensation reactions of lipid precursors in high temperature water solutions. This indicates that abiotic synthesis of organic lipid compounds can occur easily under hydrothermal conditions. If you remember, hydrothermal vents, which are just hydrothermal because they're extremely hot, they're basically underwater volcanoes. These vents represent the environment and one of the two main hypotheses behind where life first evolved on Earth. And we'll talk about these hydrothermal vents a little bit later in more detail, but basically this indication of abiotic synthesis of lipids in this environment lends more evidence to that hypothesis about where life evolved. Additionally, once lipids are formed in water, they spontaneously self-assemble into membrane-like structures because they are oh my gosh. Because they are amphipathic. This just means they have two different charges. They have a polar end called the head of the lipid, and they have a non-polar end called the tails. If you don't know what polar and non-polar means, it doesn't really matter for the purposes of this video. You just need to know that the polar heads like water and the non-polar tails don't like water. Anything that likes water, meaning it wants to dissolve in water, can easily dissolve in water, is called hydrophilic. Hydro, water, philic, loves. Anything that is non-polar, that does not want to dissolve in water, hates water, and it is hydrophobic. Hydro, water, phobic, fear of. So the fatty acid tails of these lipids will turn in on each other and try and get away from the water, whereas the heads like the water and don't mind facing outward toward the water. So this double layer structure easily forms anytime lipids are in water, as we can see easily in our kitchen. If we just pour some oil into water, it'll form these spherical things that don't want to mix with the water. That's because the hydrophobic parts of the lipids, the fatty acids, do not want to mix with that water. And that's what's forming these membrane-like structures. And this is the way that membranes likely abiotically synthesize themselves once lipids had formed. Moving on to nucleic acids. Nucleic acids are RNA and DNA. This is the informational genetic material in our cells that codes for our proteins, which are the building blocks of basically everything in life. And so this informational material is really important. And RNA and DNA have been the most difficult biomolecule class to actually produce abiotically. Their components are things we've already discussed. If we look at the picture on the bottom right, we see the structure of a general nucleic acid, which includes a phosphate group, which we can easily find abiotically in nature. It also includes a sugar, which we talked about on the previous carbohydrate slide, and how ribose could form due to the foremost reaction. And ribose, by the way, is why RNA starts with an R, ribonucleic acid, and DNA starts with a D, deoxyribonucleic acid. And the last component is nitrogenous bases, or nucleobases, adenine, guanine, cytosine, and thymine. And some of these bases were actually some of the byproducts of Miller's early abiotic experiments. So all of these components can be pretty easily explained away to have abiotically synthesized in relatively simple reactions on early Earth. However, having the components come together in the way that they are in RNA and DNA, and having the strands of RNA and DNA form, is harder to explain away. So how did these components build strands of RNA and DNA? There is a hypothesis called the RNA world hypothesis, which came about after the discovery of ribozymes, which are a type of RNA that can act as genetic code as well as catalyze reactions to bond proteins. Because of this dual behavior of these ribozymes, they have been hypothesized to be the key component in early life, leading to the first life forms on Earth that had single-stranded RNA, likely enclosed in a lipid bilayer membrane, which used simple carbohydrates to store food and replicated via ribosomes, which also played the role of proteins until it replicated enough that different proteins evolved. And so this dual behavior of ribozymes is really important for the origin of life on Earth. However, there are other hypotheses such that simpler nucleic acids came along before RNA and DNA, and then life evolved into using RNA and DNA later on, and then that dominated and the other life forms went extinct. However, it's still heavily debated, and so I encourage you all, if you're interested in this topic, to go and do your own research on it. It's really, really amazing and flabbergasts me how much we know, but also really excites me how much we still don't know about the origins of life on Earth and how many discoveries are still yet to be made. So now that we've talked a little bit about how biomolecules might have been made on early prebiotic Earth, we can move on to where it all happened. The two major hypotheses, like I mentioned earlier, include the warm little pond and the hydrothermal vent hypotheses. However, we're only going to be focusing on the hydrothermal vent hypothesis today because this has the most recent evidence that backs it up rather than the warm little pond. However, I do want to stress that the warm little pond hypothesis that occurred at the surface of Earth still has a lot of evidence behind it, and if you want to know more about it, I encourage you to read more about it on your own. But today in this video, we're going to be focusing on the hydrothermal vent environment for the origin of life. Evidence suggests that life began on Earth around 3.5 to 4 billion years ago. During this time, the atmosphere and oceans had negligible free oxygen, creating an extreme reducing environment on Earth at this time. So life evolved without oxygen. So what did it use to get energy? Well, on Earth today, we have phototrophs, which use light to gain energy, and we have chemotrophs, which use chemicals to get energy. At hydrothermal vents, or underwater volcanoes deep in the ocean, chemosynthesis is the only way to get energy because there's no sunlight penetrating at those depths in the oceans. So there's a hypothesis called the iron sulfide world, where hydrothermal vents called black smokers spew out vent fluids rich in iron sulfide compounds, which can act as catalysts for life because they have charged crystal surfaces which attract organic molecules and then can form long chains by condensation reactions by these organic molecules building up at their crystal surfaces. Additionally, they had energy in the way of heat from the volcanic vent fluid, and then you also were protected from heavy meteorite bombardment at the surface of Earth because you're down deep in the ocean, and you were protected from intense UV radiation which was occurring at the surface of Earth because there was no oxygen to form an ozone layer on Earth yet. And so Earth was receiving a lot of intense UV radiation that likely would have been hard for life to cope with on Earth's surface at this time. The last piece of evidence listed on this slide for the iron sulfur world hypothesis of life occurring at a black smoker is that archaea, which are really primitive forms of life on Earth today, are found at these vents, and they likely represent the first domain to evolve on Earth out of the archaea, bacteria, and eukaryote domains. So some scientists postulate that their abundant activity at hydrothermal vents is evidence that life began in these systems. However, there's not only black smoker hydrothermal vents. There's also what we call white smoker hydrothermal vents. These white smokers, instead of spewing out 400 degrees Celsius acidic vent fluid like black smokers do, they spew basic vent fluid around 45 to 90 degrees Celsius. This is still very hot vent fluid, but not nearly as hot as the black smokers, and instead of being acidic, it's basic. Additionally, a lot of scientists think that the really high temperatures at black smokers are unlikely to have hosted such sensitive early life on Earth, and rather that the temperatures and pH at white smokers are more likely to have hosted the first life. These white smokers are also favorable because they create energy gradients that life can harness because of the pH difference of the alkaline vents coming in to mix with the more acidic ocean water at the time. We know that oceans back during this time were more acidic because there was more CO2 content in them. And so this difference in pH led to the possibility that cells could have pumped protons across their membrane, creating a proton gradient across their membrane. And this proton gradient created what's called today the proton motive force, which is used to make ATP. This pumping of protons and creating a proton motive force to create ATP, which is biology's form of energy, is used in all life forms today. So the fact that this would have been the perfect environment for the proton motive force to be invented is a really strong evidence for this white smoker hydrothermal vent life origin hypothesis. Lastly, in addition to the protection from UV radiation, heat and proton gradients for energy, iron sulfide catalysts, and the presence of primitive archaea, there's also abiotically produced methane and formate, which are simple hydrocarbons, and redox gradients at these vents that provide further evidence that life could have gotten its start in hydrothermal vent systems. Methane and formate are used in simple biological mechanisms today by organisms that live in these systems to get energy through metabolism, so it is likely that the first life could have also been using these compounds. Additionally, redox gradients, which are just electrochemical gradients, so basically a difference in electrons, which also create energy similar to the way we talked about proton gradients creating energy, and this could have also been harnessed by life to gain energy. And so all of these components that we've talked about for hydrothermal vent systems are extremely interesting and compelling lines of evidence that life evolved in these vent systems. So now that we've discussed how the four classes of biomolecules likely abiotically synthesized and where life possibly got its start, let's talk about the categories of life. Like we already mentioned, there are three domains: bacteria, archaea, and eukaryote. We're eukaryotes, which are more complex cells, as we can see in this figure in this slide. We can see that these eukaryotes contain a lot of other things within their cell, and these other things are called organelles. However, prokaryotes, which include bacteria and archaea, do not contain organelles. They're basically just a capsule which contain genetic material, RNA and DNA, that is enclosed by a cell wall. So how did prokaryotes become eukaryotic? This seems like a huge jump from no organelles to a bunch of organelles. How did the first organelle evolve and why? So to explain this, we must talk about symbiosis. Endosymbiosis is a theory that was first proposed proposed by Lynn Margulis, who was a scientist who proposed that all the organelles and eukaryotes were once individual organisms, so basically larger cells ate smaller cells, and eventually those cells within the larger cells evolved to work together with their host cell, and it became a mutually beneficial relationship that evolved further and further until eukaryotes had a whole host of organelles living inside of them that were now part of their organism. This proposal seemed far-fetched for many years, and it wasn't until years later that we had the technology to really prove this theory. The evidence for this endosymbiosis theory is the function of photosynthesis in chloroplasts. Chloroplasts are thought to have come from cyanobacteria, which were the first photosynthesizers on Earth, and the fact that chloroplasts perform photosynthesis in all plants today is really strong evidence for the fact that they came from cyanobacteria that had just been eaten by a larger cell. Additionally, organelles like chloroplasts and mitochondria have their own membranes, make their own proteins, are susceptible to antibiotics, and have their own reproductive mechanisms. But the really clinching evidence here, which came with our improved technology, is that these organelles have their own distinct DNA that is coded differently from the eukaryotic cell's DNA in the nucleus. And so the fact that mitochondria and chloroplasts have their own DNA, along with all their other characteristics, is overwhelming evidence for the fact that these were once their own individual prokaryotic organism. So what about the timing of the evolution of these three domains? When did the prokaryotes come along relative to the eukaryotes? How long did it take for the eukaryotes to decide to start eating the prokaryotes and become a different cell type? Well, now we need to look into the fossil record to find evidence of the timing of these evolutionary events and to try and piece together the evolution of these three domains. The earliest fossils on Earth that are non-negotiable fossils are about 3.4 to 3.5 billion years old, and these fossils are stromatolites. Stromatolites are just fossilized microbial mats. These microbial mats were formed by cyanobacteria. We talked about those on the last slide being what eventually became chloroplasts. These cyanobacteria were photosynthesizing organisms that formed these huge bacterial mats, and these could easily be preserved. However, this was not the first life on Earth, and there is evidence for biochemical signatures that suggest life long before cyanobacterial stromatolites were forming. An example of this biochemical evidence comes from the Isua Formation in Greenland. This formation is about 3.9 billion years old, and it includes graphite with carbon isotopic signatures indicative of photosynthesis. If you want to know more about how we know isotope ratios of carbon are indicative of photosynthesis, you can watch my carbon isotope video. So what about the first eukaryotes? We just talked about some bacteria and some biochemical evidence for photosynthesizing bacteria, but we haven't discussed eukaryotic evolution yet. The first eukaryotic fossils are seen in preserved cells and in biomarkers. Preserved eukaryotic cells are recognizable because they are too large to be prokaryotic cells. Fossilized eukaryotic cells are found in rocks about 1.75 billion years old. However, biomarker evidence, or biochemical signatures, for eukaryotic life is found in rocks as old as 2.7 billion years. And so it's likely that eukaryotes evolved long before 1.75 billion years ago, but maybe they weren't that extensive, or they just weren't very easily preserved. However, the picture down here to the bottom left shows one structure that only eukaryotes form that is really robust and is typically preserved in the geochemistry of the rock record. So what about more complex life? Up until this point, we've only discussed single cellular prokaryotes and eukaryotic life. But what about something with multiple cells? Something a little bit bigger? Something that maybe we could see with our own eyes? Well, it's not until we see the Ediacaran fauna, which evolved around 600 million years ago, that we actually get abundant multicellular life in the fossil record. You may not think that the few localities that these organisms are found in calls for the term abundant, but it does. Soft-bodied organisms very rarely preserved, so the fact that there are a handful of fossils at all means that these guys were really abundant during this time, and therefore evolution was starting to ramp up into multicellular life. And as we see on the bottom of the slide here, these soft-bodied invertebrate animals were similar in morphology to sponges, worms, soft corals, and other simple soft-bodied organisms. And just after the Ediacaran fauna evolved at around 550 million years ago, the Cambrian period began, which began the evolution evolutionary radiation or biodiversification called the Cambrian Explosion. This is the time that marks the first appearance of hard parts or skeletons, shells, teeth, etc. And these hard parts were really important for preservation. But why do we call this the Cambrian Explosion? Was it really that evolution was occurring much faster than it did at any other time in Earth's history? Well, not really. This name explosion is actually kind of misleading. It wasn't really explosion as much as it was kind of a Cambrian slow fuse, which is definitely not as exciting, but it's more accurate. While the Cambrian does mark a period of evolution in which things evolved that hadn't shown up in the fossil record before, it's not that it's happening extremely fast, it's just that it's happening at a normal rate for evolution, whereas evolution before the Cambrian period was occurring very slowly. So the question that we really should be asking is, why did it take so long to get multicellular life, and why did evolution before the Cambrian occur so slowly? Well, the answer is found in a multitude of evidence. The first line of evidence is oxygen content. Like we talked about earlier, on early Earth there was not much free oxygen. It was pretty much negligible. However, at around 2.4 to 2.2 billion years ago, oxygen content rose in the atmosphere and oceans due to something called the Great Oxidation Event. I talk about the Great Oxidation Event, or the GEO, in my Great Oxidation Event video if you want to look more into that. And this event likely led to the first eukaryotic life. However, like we talked about before, that eukaryotic life was still single-celled until way later in Earth's history, close to the Cambrian. So why did it take so long for those eukaryotes to become more complex? Well, there was still very little oxygen in the Earth's atmosphere after the GEO compared to today's atmosphere. It wasn't until just before the Cambrian, as we see here on this graph, that oxygen jumped up once again in another oxidation event, increasing the oxygen levels in the atmosphere from about 10% of today's levels to about the current level of oxygen in the atmosphere today. This second jump in oxygen likely gave the eukaryotes that extra push they needed to be able to begin oxygen respiration, which is the most highly energy-yielding metabolic pathway of all other redox pathways that life utilizes to gain energy. And this is likely what led to more complex life. Additional reasons for why it took so long for complex multicellular life to evolve and why it seemingly occurred suddenly in the Cambrian is because following the snowball Earth event at around 700 to 600 million years ago, global warming occurred, creating new warm shallow sea environments and causing the transgression of shallow eperic seas onto continents, creating prime evolutionary niches for biodiversification to occur. Additionally, rapid tectonics increased nutrient supply to the oceans, leading to calcium phosphate skeletons, and later, as oxygen content increased in the atmosphere, calcium carbonate skeletons, and this ability to grow skeletons, shells, teeth, etc. really opened the door for biodiversification even more. Additionally, you had the cropping hypothesis, in which organisms such as certain types of mollusks evolved to graze on the cyanobacterial mats and algal mats that covered the warm shallow sea floor environment in the late Precambrian, and this cleared the ocean floor, creating a whole new world of perfect surface and subsurface niches for even more biodiversification. All of this diversification that occurred in the Cambrian led to a multitude of invertebrate animal phyla evolving. And these phyla are what we're going to be going over in the paleontology playlist, or at least the invertebrate paleontology playlist, in which we talk about the porifera phylum, or sponges, which have multicellularity and are animals, however have no true tissues. Additionally, we talk about the cnidarian phylum, which have tissues and radial symmetry. Then we talk about the mollusca and brachiopoda and bryozoan phylums, which have bilateral symmetry and body cavities. And then we talk about arthropoda, which have segmentation, molt, and have jointed appendages. Then we talk about echinodermata and hemichordata, which have all those traits, as well as being classified as deuterostomes, and we'll talk about what all that means in those phyla videos. And so I just wanted to show this cladogram where you can see all of these phyla related to one another and how they relate to one another, as well as what traits they have, so that now you hopefully feel prepared to watch all the rest of the videos over these phyla. Lastly, we will likely talk about the chordata phyla down the road, maybe in a whole different playlist, I'm not sure yet, because that's getting into the vertebrates, which is a whole nother game than invertebrate paleontology. But anyway, for this video, that is all I have for you guys. I hope that was helpful in learning a little bit about the origins of life on Earth, and possibly even other planets and moons, because there are other planets and moons that also have hydrothermal vents. So it's really cool how applicable this is to maybe finding life on other worlds. So anyway, If you want to look at the rest of the playlist for paleontology, you can click down below. Thanks so much for watching, and I'll see you guys next time. Bye!